I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
So that said, welcome to our December, National Roundup brown bag session. We started these in May when we first were wishing we could meet each other in person and wanted to get research shared. And we've been having them all year, focusing on Wisconsin research. We thought for December we'd want to have a little broader reach and be able to hear from some of the other exciting research that's going on in cranberries throughout North America. And Extension, University of Wisconsin-Madison Division of Extension has been, providing me to help out with these. And the Wisconsin State Cranberry Growers Association has been kind of the leader and champion of the idea. So I want to give Tom a minute to say hello to everyone.
Well, good afternoon and season's greetings, however you're going to celebrate the holidays and the end of the year. We hope you enjoy it and do it in a safe and, somewhat healthy way. But first of all, just thanks to Allison for organizing this. Her and Alex have worked very hard on this. This is our final brown bag of the year. One of the things that we've learned through this process is that while we all would like to be meeting in person, this does gives us the opportunity and the flexibility to provide some quality programming to people, not only here in Wisconsin, but across North America, which is what we're trying to do today. We thought we'd invite people in from the other growing regions to hear the information as we expand to learn more about what's going on outside of our state borders, and as my good friend Brian Wick referred to it as shrinking the industry. So this is something that we're going to be continuing into 2021 and beyond. It's a great tool for us to use. It never replaces getting together face to face, but it does provide the opportunities to exchange some information over an hour without a lot of travel. So we're glad you could join us today, and we thank Allison for putting it together. Just one quick reminder is that the Wisconsin Cranberry School will also be virtual this year, and we'll be conducting that on the 27th and 28th of January. If you're interested in attending, I know information has gone out to Wisconsin growers, but if growers from other regions want to join us and learn more about the program, they can contact the Growers Association office or get a hold of Alex Skowinski or check our website out at www.wiscran.org, and you can get the information for registering and participating. So with that, thanks to everybody for joining us today, and I'll turn it back over to Allison.
Thank you. So with an awful lot of really exciting cranberry research going on throughout the world, we wanted to have a lot of information, but still wanted to give people enough time to speak meatily. So we've got three people here with us today. We've got Peter Jaranyama from UMass, Cesar Rodriguez-Saona from Rutgers at the Mariucci Center, and then Jim Polishok at the USDA ARS. Next up will be Cesar Rodriguez-Saona. So he has been working in general on kind of secondary pests, and we've heard from him earlier this year on blunt-nosed leafhopper, which is a topic of a lot of interest in Wisconsin this year. And continuing, of course, if people have questions, feel free to type them either to the whole chat or directly to me, and I can ask on your behalf.
So thank you, Allison, for the invitation. And so today I'm going to be talking about the work that we have been doing on blunt-nosed leafhoppers in New Jersey. I know that this is becoming a concern in Wisconsin also. And before I start, I want to acknowledge my technicians, Robert Holcraft and Vera Kirishenko-Roth, who conducted most of this work that I'll be presenting today. So this is just an outline of what I'll be talking about. I'll just give you a brief biology of leafhoppers, and then talk about monitoring. It's very important for growers to monitor for these pests. And then I will talk about some of the trials that we have been doing to look at different insecticides and their efficacy against blind-nosed leafhoppers. So blind-nosed leafhoppers are native to the US, same as cranberries, of course, and they are distributed throughout the eastern states of the country. And they feed on several host plants, and they are mainly a problem. They do feed on the plant, they feed on the phloem, but they are mainly a problem because they transmit bacteria. It's a phytoplasma. It's a type of bacteria that causes false blossom disease. And this disease is characterized by the malformation of flowers, causing the flowers to stand erect. And also you see changes in color in the petals. They become redder than normal. And because the phytoplasma in infected plants are alive, but they are sterile, and their only purpose is to vector the disease, they are commonly referred to as zombie plants. So this disease impacted the cranberry production in New Jersey 65 years ago. The production of cranberries in New Jersey was much greater than what it is today. And growers attribute this decline partially to false blossom disease. And in the past 2 decades, we have seen an increase in leafhopper populations in New Jersey, mainly due to changes in pest management practices. Our growers have increased the use of low-risk, target-specific insecticides that mainly target lepidopteran pests, such as Aspergillus or spotted fireworm, such as Intrepid, Delegate, and Altacore, and reduce the use of broad-spectrum insecticides, particularly before the bloom. So blind-nosed leafhoppers, they have 1 generation a year. They go through 3 stages: the eggs, the nymphs, and the adults. They have 5 instars during the nymphal stage. They overwinter as eggs, and in New Jersey, the eggs hatch in May. The adults are most abundant in July, and they lay their eggs in July and August. So as I mentioned, monitoring is very important. And we do that using sweep net sampling before bloom to monitor for the nymphs. However, the nymphs are very difficult to monitor because they are fairly small during the time of the year, requiring the use of a microscope. So you have to collect the samples, place them in a bag, and then take them to your lab or office and look under the microscope to count the number of nymphs. So to make it easier, we did a study with Dan Schiffauer, former Ocean Spray, and to look at whether we could use different color sticky traps to monitor for the adults. There's no trapping tool to monitor for the adults currently available, and monitoring for the nymphs using sweep net can be labor intensive. So we tested these different color traps to monitor 2 species that are common in cranberries in New Jersey: the blind-nosed leafhoppers and the sharp-nosed leafhopper. The only species that is known to transmit false blossom is the blind-nosed leafhopper. But we use yellow sticky traps to monitor the sharp-nosed leafhoppers in blueberries. So we thought that this could be applied also in cranberries. So we tested 6 colored sticky traps to monitor the adults from June through August. And we found that the sharp-nosed leafhopper are strongly attracted to yellow. As we expected, this is the color that we use to monitor sharp-nosed leafhoppers in blueberries. However, the blind-nosed leafhoppers were mostly attracted to green and red, but their attraction was not very strong. which makes the use of these color traps to monitor blindness leafhoppers not very reliable.
Those are
Yeah.
total counts as opposed to I mean, did you do sweep nets in addition to have a comparison of how many were attracted to the trap versus how many were present total?
That's a good point, Allison. There were a lot of, we picked these beds because they had a lot of leafhoppers. The infestation was very high. And there was a correlation between the numbers that we were catching on the traps and on the sweep nets, but we weren't catching as many as we would have expected. Especially like if you look at the numbers were fairly low, and these are like, you know, averages. So we were expecting to see more on those traps.
So there were plenty that were
based on
not
the
being
numbers that
attracted. We didn't have complete. Okay.
Exactly.
Thank you.
Exactly. So so in the past two decades, we have been testing several insecticides since I joined Rutgers in 2005. So we tested different insecticides with different mode of actions. And in this first trial that I'm showing you, this is was done in 2014, fairly recently, where we tested Exirol. We tested 2 unregistered insecticides and Lorsban. And the way that we do this test, we treat plots in cranberry beds at the Marucci Center using a backpack sprayer, and then collect the foliage from the treated plots. And we bring the foliage to the lab where we place them in these water picks, and we cover the foliage with, we place the foliage inside the vials, where we release a certain number of leafhoppers inside the vial, and then we look at the mortality. We do this because a lot of the insecticides that we use are not registered. So this is a way to screen through insecticides that are unregistered at the moment. So what we found is that we got suppression, like a reduction in the numbers of leafhoppers with Closer, the 2 unregistered insecticides, and Lorsban as a standard. So in another trial that we conducted, just looking at Closer and Lorsban at different rates, we found that this is the percent mortality. The one previously is the percent survival. Sorry about that. So you know, the smaller the number, the better. Here, the larger the number, the better. So this is percent mortality. You can see that Closer and Lorsban provide a good control. Unfortunately, Closer is now registered, but unfortunately, it's prohibited for use before bloom or during bloom, of course. So the only time that you can use Closer is after bloom, which limits its use against leafhoppers. So in another trial, we tested Actara, Acel, 2 neonicotinoids, Movento, and also an unregistered insecticide, compound B. And as you can see, the neonicotinoids are fairly good against leafhoppers. However, we don't recommend in New Jersey the use of neonicotinoids before bloom because of potential negative effects on bees.
I want to remind people to, as we're getting through these results, go ahead and type questions into the chat so I can ask questions to Cesar at the end.
So we did also a larger scale trial to mimic more what a grower would do in a normal condition, where we tested Movento in this case in larger plots. And we found some suppression with Movento. Movento is a growth inhibitor. So you have to like apply it early in the season in order to get some effect. It's not going to be very effective against adults. So now Movento is registered, but again, its use is limited to after bloom. So that again limits its use against leafhoppers. So in 2018, we did we screened a larger number of insecticides. So we had Leaf, Closer, Acel, Agrimec, another unregistered insecticide, a pyrethroid that is under IR4 currently, Lorspan 7, Cormorant that is a mix of Ryman and Acel, Verdaprint that just got registered is a new diamide, and Movento. And we did the same type of experiment looking at toxicity against the nymphs and adults. And this is the data for the nymphs. We found, again, Closer, Acel, Cormorant, the ChemC that is an unregistered pyrethroid, Lorspan, and 7 provided good control. The adults were less susceptible, but you can see ChemC, Lorspan, and 7 were the most effective. Closer had some effect, but you can see that it was not different from the control. The problem, again, of Cormorant, some of these newer insecticides that they are not allowed to be used before bloom, and definitely not during bloom. So Cormorant can only be used after bloom, which limits, again, its use against leafhoppers. So, so in conclusion, right now, the best way to monitor for leafhoppers is to monitor before bloom for the nymphs. And the best options that you have for treating the leafhoppers is to treat for the nymphs before bloom with Lorsban, diazinon, and 7. So some of the recommendations that I'm giving to our growers is they always ask me about the threshold, an economic threshold. And it's difficult to establish an economic threshold since the proportion of infected individuals may vary depending on the variety. So as an IPM practice, as I mentioned, we recommend to monitor blind nose leafhopper populations before bloom using a sweep net. And if the populations are increasing compared to the previous years, or the beds had not been treated for blind nose leafhoppers for 2 to 3 years, growers should consider treating their beds. And we have established some tentative thresholds. I have been in discussion with some growers on how they feel like would be a good threshold for leafhoppers. And we're coming up with 20 nymphs per sweep net in young beds, new variety beds, and or beds that had incidence of false blossom. And a higher threshold of 40 nymphs per sweep set if the beds are older varieties, and the bed doesn't have any history of false blossom. So in most cases, there is no need to apply a broad spectrum insecticides to control blind nose leafhoppers for 2 or more consecutive years. So if you apply 1 year, you control the leafhoppers, you don't need to apply the following year. And as I mentioned, we don't recommend the neonicotinoids like Acel or Actara before bloom because of their potential negative effects on bees, although they are effective. And Closer is also effective against nymphs, but is prohibited to use before or during bloom.
So we have a couple.
So what are, yes.
I was going to say
this is my last slide, and I can go with the questions. So what are things I feel like is needed? So so I feel like we need to learn more about what are the landscape factors that are influencing the distribution of leafhoppers in cranberry farms. Like we don't know how well they are distributed and how many beds we need to treat in order to control it. We don't know also what's the proportion of leafhoppers that are infected with the phytoplasma across different beds and in different varieties, especially considering that some varieties, like some of the new varieties, might be more susceptible to the infection or more susceptible to leafhoppers. We also need to know more about the landscape factors that are influencing the levels of leafhopper infection across beds. And we need to develop more sustainable methods to control leafhoppers and reduce false blossom incidence. So, Allison, I'll take the questions.
This has been, fantastic, fantastically important. And as one of the questions had asked, Has false blossom been found in commercial beds in Wisconsin? Yes, it has this year. We've since we had this resurgence of blunt-nosed leaftopper, people have sent in samples of vines that they wanted checked to our plant pathology lab at the University of Wisconsin. They have a very good assay for checking vines for the presence of the phytoplasma, and we do have it confirmed in a few commercial beds. We're working on having an assay for, does this blunt-nosed leafhopper carry the phytoplasma? That's still kind of being developed, and that's something that is an interesting ongoing talk. I don't know if false blossom is spreading in New Jersey and Massachusetts right now, but maybe Cesar has a word on that.
False blossom, is it spreading in Massachusetts, or?
Is it spreading in Massachusetts? Is it spreading in New Jersey? Do you know?
I know that it's in Massachusetts, and that's something that, leafhoppers are common in natural habitats in Massachusetts. They tend to spray more than we do in New Jersey. So they don't tend to see as many populations inside, like in commercial beds. The one reason why we see leafhopper numbers, higher leafhopper numbers, as I mentioned, is because we have reduced the amount of broad spectrum insecticides. So growers in our state, they only use like 1 or 2 insecticide applications a year, and they tend to use some of the newer insecticides. So that caused changes in the pest complexity. Not just leafhoppers, but also toad bugs. We have also problems with cranberry toad bugs in some areas. Okay. And that was also a result from these changes in pesticide use.
So I have 2 more questions. One, I'm going to combine one, and the other is kind of a standalone. Did you test any biological insecticides for efficacy? Iceria fumorosea or Bovaria basania?
No, I haven't tested any biologicals. I mean, that would be great. The one insecticide that I'm going to be testing is danitol next year. It's registered now, but we don't have any efficacy data. And I don't know if also Ocean Spray is going to be recommending it. We need to get MRLs. I don't think we have MRLs for that one. There's also another candidate, another new insecticide that has been registered in blueberries, but not in cranberries, that has effects on piercing sucking insects. So this morning I contacted the company to see if there's interest in also extending the label to cranberries. But yes, I haven't. Okay. I would love to collaborate with the researchers in Wisconsin and work on some of these things. As I mentioned, at the end, we need to know more about the incidence. How many leafhoppers are infected with the disease across different varieties, and because we see that in some varieties, only a few of leafhoppers can transmit the disease. You don't need that many. So the threshold, that's why the threshold for us is lower in some of the newer varieties.
And I want to get this one final question out, and then we can move along to Jim. The question kind of combined, someone asked, have you seen false blossom in Stevens, or is Stevens still resistant to it? Someone else mentioned that years ago, resistance of cultivars was more preferential non-feeding by the leafhoppers rather than disease resistance. So maybe the phytoplasma is equally effective against all cranberry varieties, but the leafhoppers prefer some varieties to others. Are your new hybrids showing any resistance either to the disease or to feeding by the vector?
So that's something that that we were planning on doing this year. We had plans to look at resistance across different varieties against the nymphs, and we couldn't because of COVID. But we are planning to do that next year. Yeah, the mechanism of resistance, there's definitely resistance. The mechanism We don't know. We looked at infected plants, and leafhoppers like infected plants. The infected plants have more nitrogen, and it benefits the vector. So the vector seems to spend more time on infected plants than non-infected plants, which may help the transmission of the disease. We didn't find any smell, any particular smell from infected plants that attracted the leafhoppers. So we think that the attraction is mainly by visual cues. So they definitely look different. Mm-hmm. You can even you can see the differences just by looking at the bed that has the infected.
I'll just take Frank's last comment here. He says false positive may be similar to Lyme disease, where a certain % of the vector possesses the pathogen. That's definitely the case. Not every blunt-nosed leafhopper has the phytoplasma in its system. It has to first, be born, feed on an infected plant enough so that it has a level of the phytoplasma built up in its fluids. And then when it feeds on the next plant, it's able to transmit. So by default, when a new blunt-nosed leafhopper is born, it's not definitely transmitting the phytoplasma. It has to feed and obtain that phytoplasma before it can transmit it.
Yeah, it's a persistent. Yeah.
Yes. So thank you so much, Cesar. That's been very interesting. We've, of course, had a lot of questions this year as we've seen new blunt-nosed leafhopper resurgence in Wisconsin, and it's good to know what's going on in the rest of the nation. Thank you for presenting.
Thank you. Thank you for the invitation.
Then Jim, we got any other questions that we want to get to while we're still in recording mode? I appreciate people holding with us through the 1 o'clock hour, but this has just all been so fascinating. Peter and Cesar and Jim, all three, and of course all of the institutions that you represent, all of the growers throughout the hemisphere, taking advantage of this knowledge. So any further questions? I will go ahead and stop the recording then, and thank you guys all for joining. Thanks again, as always, to the Wisconsin State Cranberry Growers Association. If you're interested in being on my invitation list for these going forward, feel free to email me. I've got my address there, allison.jonjak@wisc.edu, and look forward to seeing you in the new year, and have a wonderful Christmas and upcoming Cranberry School.